We're going to get started on our different tissue types. So really the first tissue type we're going to get into is what we refer to as epithelial tissue. This is the tissue that is really open to the outside everywhere, probably one of the easiest ones to identify in terms of that the cells do kind of tend to look a little bit similar to one another even in the different types. So epithelium, we're going to see, is going to have a number of characteristics and serve a few different functions, and that's, again, one of the things that we're going to be talking about. With a lot of these tissues, especially on the introductions, a lot of this is going to be about what is the type of tissue in terms of the name. When we talk about that, what does it mean it looks like? What are the characteristics of this tissue? And what is it going to be good at? And then because of those things, where might you find it in the body? So what does it look like? What does it do? Where do you find it? Those are kind of the key ideas a lot on these intro chapters on this stuff. So if we're looking at epithelial tissue, like I said already, it covers the surface of the body, it lines cavities, it makes glands. So it's going to do a number of different functions. And again, to me, thinking about the different types as we start describing them, out of these different functions, what might that particular type be good at is a good thing to think about, because then you can start almost inferring places you might or might not see it. So functions of this are going to include a number of different things. Obviously, if it's making up glands, it's doing a lot of secretion and absorption type of stuff. Another thing that epithelium does, it's mainly certain types of it, but it's really involved with protection. Again, it's lining the outside of the body. That needs to be protective. And then the fifth option here, filtration, or if we wanted to say diffusion, osmosis, allowing those things to happen quickly, that's another big function. The other thing with sensory reception, a lot of the nerves that are going to house that are found within epithelial tissue. But again, epithelial tissue is found pretty much everywhere. You can see on the picture on the right, the epithelial tissue is going to be found a lot in the glands here, up on the surface up here, making up some of these glands that are going with the hair follicle. Those are going to be all things that are made up of epithelial tissue. So these are the things it does. And one of the things, so the main functions of it here, the other thing we're going to talk about is characteristics of epithelium. One of the things I don't want you to get confused with, if I talk about, give me 3 characteristics of epithelium that makes it unique, don't give me the functions of epithelium. Make sure you're giving me the characteristics, and vice versa. So certain characteristics of epithelium, one of the ones, and probably to me the hardest one to understand, is this idea of polarity. The main thing with epithelial tissue is it is going to have it has a top and a bottom. It is directional. The main thing on these ones, different areas might have different proteins on the surface, so that's one of the ways we talk about polarity. The other thing, there is a bottom side called the basal side. This is going to be closer to this, what we call a basement membrane. So the blue arrow over on the right there is showing that basal side. The side that's open to the space or open to the lumen is what we refer to as the apical side. And again, all epithelium is going to have this polarity. It's going to have a basal side, it's going to have an apical side. And again, what the shape is and things like that on the apical side are going to be, a lot of times, help you determine what type of epithelium we're talking about. I mean, it's also polar in that there is a lot of times certain proteins that are only expressed on the apical membrane, certain ones that are only expressed on the basal membrane, and sometimes stuff might be on the lateral side too, but there is some polarity in terms of protein expression some of the time. Another characteristic of all epithelium is that it has a layer of connective tissue underneath it known as the basal membrane or the basal lamina. This is the arrow on the right there again, the different arrows there pointing at that basement membrane. This is that connective tissue that's found under all epithelium. Like I said, any place you see epithelium, it will have a basal membrane underneath. So again, apical is that lumen side. So in the case here, this would be the apical side that's facing lumen, not necessarily the outside. If you're talking about the skin, this would be the apical surface. Basal surface is going to be that underneath, closer to the basement membrane. Basolateral, lateral is going to be on the sides here. We're going to see a lot of this in the next little set of ones here. That lateral side is really tightly connected by these tight junctions and adherens and things like that. So that basal region, like I said, the basement membrane is made up of these two layers called the basal lamina and the reticular lamina, again, made up of different types of connective tissue, things like different types of collagen and laminins, fibronectin, things like that are all what are making up this basement membrane. To me, I'm not so worried about you differentiating the basal lamina and the reticular lamina, but the main thing is some of this is made by the epithelial cells, some of this is made by the connective tissue underneath the epithelium. But again, this basal lamina or this basement membrane you can see is underneath everything right here. We can stain for it specifically with certain types of stains. And again, there is these things called hemidesmosomes that are going to help hold this to that layer underneath. But again, this is what is anchoring that epithelium to the underlying tissue. The other thing is when we talk about epithelium, another characteristic is and I always kind of joke around, and it sounds funny to say this, but it's very cellular. The main thing with epithelial tissue, it is really defined by the fact that it's a bunch of closely packed cells to one another. Again, a lot of these are protective. Having all these cells butted right up to one another gives us a nice continuous layer. The main thing is when we get to connective tissue, you'll see a lot of times connective tissue is sometimes defined by the space between the cells and what is in that space, where epithelial tissue is mainly all cells with a basement membrane underneath. The other thing about these cells is they are tightly connected to one another. Again, especially for these multi-layered ones that are going to be protective, that makes a lot of sense. You want cells, I always kind of say a brick wall is protective, but if you have a bunch of holes in that brick wall, it's not particularly protective. So we want these cells to be continuous. We want them to be tightly connected so we can't just pop a cell out and have something get through. So these are held in place by these what are called tight junctions and desmosomes. So again, these are going to be on that side here between these epithelial cells. A lot of times I refer to these as these terminal bars, these areas in between here. You can stain for that, but again, it's a combination of these zona occludens, zona adherens, and these desmosomes. So you get almost like this ring as well as these spot welds that are going to be holding these cells together. So you can see the zona occludens up here, closer to the apical surface, the zona adherens that are a little bit deeper in, making a ring around the entire perimeter of the cell. We then have these desmosomes, which are at discrete sites that are like these little spot welds. All together, this is holding these cells together. Sometimes we will also see gap junctions that let nutrients from one cell go into the neighboring cell. But again, these cells are very well connected to one another. And you can see some of these on these electron micrographs. Again, I'm not going to give you a picture of this and expect you to identify these things. Again, this is a little bit more of an introduction to kind of what is special about these types of cells. We will be looking at the different types of epithelium as we get in a little bit further and definitely being able to identify some of those. So you can again see the different parts here. Again, all this together is help holding these cells tightly together. Again, linings, the skin on the outside of the body, if this is going to be protective, we don't want things to be able to travel in between the cells very easily. So by having these tightly connected, this is going to help maintain the integrity of that gland, of that tube, of that particular protective layer. And again, we don't want spaces in between those because that would be spots for bacteria, other things like that. Skin, a lot of times you don't get a lot of infections on your skin until you get what? A cut in the skin, which is opening up spaces between epithelial cells. And again, just kind of showing you some of the different ones here, how these can be kind of combining together. And really the last couple of things here, one of the things we'll see about epithelial tissue, and if you've ever been cutting something on a cutting board, let's say, and you kind of nick yourself and you look down, oh, no blood. That means you just cut your epithelium. So epithelium, one of the things we don't have in it is blood vessels. It is avascular. Even the multi-layered types are avascular. You can put a needle through the outer layers of your skin without drawing blood. That is because you're only in the epidermis or the epithelial tissue. Beneath that, we're going to have a lot of blood vessels, but epithelial tissue in and of itself is avascular. And then finally, it regenerates really well. Again, this makes sense, especially when we're talking about it being a protective type of epithelium. We don't want something the first time you damage it, you can never replace it. And if that was the case, it wouldn't be particularly protective. So this type of tissue unlike a lot of other types. So nervous tissue, muscle tissue does not really have the ability to regenerate well. So they might think like, oh, cells can always divide. Not really. So the fact that it has this ability to regenerate does allow it to do some of the functions that we find important for epithelial tissue. So if we look at the surface of this, we will sometimes see some specialized structures on the surface of an epithelial tissue. So one of the things we will see probably most commonly is going to be what we refer to as microvilli, but you can also see some of these specialized cilia called stereocilia, especially in some of your sense organs, and then cilia in things like the fallopian tubes and the trachea, or some of these surface apical region structures we see. So microvilli, one of the functions we talked about of epithelium was the idea of absorption. Microvilli are a bunch of these little finger-like extensions that are coming off the cell membrane. It's increasing the surface area. There's got some of these intermediate filaments going up here and holding them to the rest of the cell, providing the structure of these. But the main thing on this one is it increases that surface area. More surface area means more ability to absorb nutrients. So you see this a lot of times in areas where a lot of absorption is taking place. For example, the small intestine. Stereocilia are not cilia. These are non-motile. You see them in the epididymis. One of the places you see these a little bit more where they are connected to some of the nervous tissue is in the vestibular senses. So your static equilibrium, your dynamic equilibrium. So this inner ear, you also see these stereocilia in here. The other one thing here is cilia. These are motile, have that 9+2 arrangement of microtubules, can move these past one another. We see these in places where we want to create current a lot of times, or move a fluid or move a substance past the cells. So for example, in the trachea, you don't want anything that you inhale to always be stuck in your lungs. So you have cilia in your upper bronchi, your trachea, that are going to help push stuff back up to the throat region, move these things past the cell, like I said, kind of like crowd surfing. Same thing with the fallopian tube. When that egg cell is ovulated into the fallopian tubes, you want it to make its way down to the uterus. So the cilia is moving these fluids and that egg cell towards the uterus. So I think the last thing we talk about on this first show here is really getting into how we go about naming or classifying these different tissues. So epithelial tissue, we really classify it based on two things. It's really the number of layers is one part of this, and then the shape of cell. We're going to see outside of that, there is then some specialized types that don't fall into this naming system, but this is the main way we are naming a lot of this. So the upper first two there is you can either have simple epithelium or you can have stratified epithelium. Simple epithelium is one layer thick. So what that means is all the cells in that layer are connected to the basement membrane. Stratified means we have two or more layers, i.e. one of those layers not connected to the basement membrane. Even if the cells aren't the same height, if everything's connected to the basement membrane, it is a simple type of epithelium. So simple is a single layer, two or more layers is stratified. We can then look at cell shapes. Squamous cells are very, if you're looking at the height of them, they're much wider than they are wide, kind of irregular shaped if you were looking down at the surface of them like this, but from the side they're going to be very flat. Really the puffiest part is generally the nucleus. So the cell width of them is much greater than the height. The cuboidal cells, on the other hand, are about the same width and height. They do not have to be squared off. They can be more rounded, but they have a similar height and width. And then finally, you have what are called columnar cells. If you think about a column, it's much taller than it is wide. If you're looking at like a column on a porch. And that's the same thing with these cells. Columnar cells are going to be much taller than they are wide. So you can see here, squamous cell, you can see from the side view, very flat. Nucleus is the widest part here, where cuboidal cells, height and width are a lot more similar. Where these columnar cells, you can see here, taller than they are wide. Nucleus tends to stay towards the basement membrane a little bit more. Again, we're going to go through each of these. I don't know about tall and regal, but they're definitely taller than they are wide. So where you look at simple epithelium versus stratified, you can see here. Here's a simple squamous. You see how they're kind of irregular when you're looking down at the surface of them. But again, from the side, very thin, much wider than they are tall. One layer. So you can have simple squamous. You can have simple cuboidal, single layer of cube-shaped cells. You can have simple columnar, and it can be ciliated, so simple ciliated columnar epithelium. You can see taller than they are wide. But again, every cell is connected to this basement membrane, which makes it a simple epithelium. And again, you can see some of these in real micrographs here. So this is looking at a capillary surrounding a blood cells right here. You can see that simple squamous cell right here, much wider than it is tall. The thickest area here is that nucleus. It's kind of almost like a fried egg, we'll see. Again, you can see another one here. Again, width really wide, nucleus kind of the thickest portion here. Simple cuboidal, you can see the height and width are about the same. Again, some of these, every once in a while, you might see where it kind of looks like, okay, maybe there's two layers right here. Could be that this is just slightly angled. Again, everything what we're doing is a 2D slice of a 3D structure. So if it's not dead on perfect of a cross section, you might see what looks like more layers. So some of the time it's finding areas that show it the best. Stratified epithelium, on the other hand, is going to have multiple layers. How we decide what a stratified epithelium is, it is going to be what is the shape at the apical surface. So if we're looking at stratified squamous here, it looks almost columnar down here, cuboidal through the center, squamous at the top. Because this is the apical surface, and they are squamous shaped cells up here, this is stratified squamous. It is whatever is at that apical side. When we're talking about squamous versus cuboidal versus columnar, it's going to be whatever's at the apical side determines that. So this one, flat cells at the surface, stratified squamous epithelium. One of the weird types here is pseudostratified epithelium. So pseudostratified epithelium, you can see here the heights of these cells are different. The main thing you see is if you look at all these cells along here, they are all connected to the basement membrane. These are not stratified cells. They are actually a type of simple epithelium, but because it looks stratified, pseudo means fake, so fake stratified epithelium is pseudostratified and columnar epithelium. So that is one of those weird kind of, doesn't fit into our basic 6 of simple squamous, simple cuboidal, simple columnar, stratified squamous, stratified cuboidal, stratified columnar. Pseudostratified would be one of those types that falls out of those basic 6. So this, you can see that stratified squamous, again, can look more cuboidal down lower. You get to the surface, it's flat. That is what we're talking about. That apical surface is flat or stratified squamous. On this one, you can see it might be a little bit flatter at the bottom, but the apical surface is cube-shaped. So that would be a stratified cuboidal. So what we're going to do from here on out with these epithelium types now, we've introduced the basic idea of how we name them, what are the characteristics, what are the basic functions of this. What we're going to do now is we're going to work through the different subtypes of this and take a look at all those and then talk a little bit about shapes of glands and how they can look a little bit weird under the microscope. What I will do towards the end of one of these ones here is I will also go on to digital micrographs and actually show you some zooming in and the controls of some of this stuff here and how you can look at it on, I won't necessarily start from the Moodle page, but how you can do it from, actually maybe I will go from the Moodle page on this first one here and show you how you get into it and how you can find some of these different ones here. So that's what we'll be doing on the next show here, so I will see you next time.